Good afternoon, ladies and gentlemen, and welcome to the fourth in our series of Forest Ecology seminars. Today's speaker is Petr Baldrian, another one of those people that needs really no great introduction. Petr Baldrian is Professor of Environmental Sciences at the Charles University in Prague. He is a highly cited researcher with over 31,000 citations of his work. He's probably one of the most cited people in his field. He's, I would say, best known for his work linking microbial diversity to function. He does this at a number of different levels, which is also what he's going to talk about today. He is the editor of a number of journals. He is, last but certainly not least, currently the president of the Czech Science Foundation. He will talk about fungi in temperate forest soils, ecology, and contribution to ecosystem processes. Just before we start, I would like to remind you that this seminar is recorded, so that if you do not wish your name to be shown, then you should remove your name. Also, at the end, we will have questions. The questions we would ask you to write into the chat box, and we will take them in the order that they came and try and make a bit of structure into it. So, Petr, your floor, as it were, or your screen. So thank you very much.
Thank you. I hope that you can hear me, and I will start sharing my screen. So thanks for the great introduction. I would say that my high citations is just a coincidence of the methodology developments where I was one of the first there. It's not so much about the excellent science, I would say. But I would also say that I'm especially happy that I was invited by Mendel University and Douglas, because much of our work is actually from their area, from the Křtiny forest. You can see a picture of one of our permanent sites where we continue measuring development of fungi in time. So it's very well fitting. And last but not least, I welcome everyone. I noticed a few friends or colleagues that I know. I have to apologize that I will talk about not about the latest results, just a bit more about an overview of what to expect. I should start with thanking all my collaborators. As Douglas told, I am now a bureaucrat for 3 years, but this period in the Czech Science Foundation is ending this year, and I hope to be fully back in science since January next year, so that's great. But I had the opportunity and good luck for very nice and good collaborators all over the world. So I wish to acknowledge them now because they really deserve it. So for the forest soil, or especially topsoil, because it's sometimes complicated to go deeper with the sampling, we can distinguish what nutrients we see there, and it somehow defines what we see in terms of the microbes. So the nutrients that we can expect come either from leaf and root litter, which is a major pool. It's a major source of organic matter, mostly of a polymeric nature, cellulose, lignin, other polysaccharides or proteins. And typically numerous enzymes are required for degradation because it's complex. Then we have another source, root exudates, nutrient source in mineral and organic soil, more readily available carbon. But within root exudates, I would also count the carbon that is passed to mycorrhiza directly in the root without being exuded. So let's say that this is this fast carbon that goes there, and you may know that this flux of carbon is very important. Then the microorganisms take the different share in this nutrient ecosystem. Obviously, in addition to carbon, there is mineral nutrients, but there the situation for most of them is the same. But for the carbon, the saprotrophic fungi are specialized in obtaining carbon by organic matter decomposition, then produce extracellular enzymes, and they are able to cope with the complexity mycorrhizal fungi don't need to care so much because they obtain carbohydrates directly from symbiotic plants, and in exchange they provide certain nutrients to plants. So if there is mycorrhiza, the nutrient demand goes for mycorrhiza, but also through them to the trees. And saprotrophic abilities vary between mycorrhizal fungi. I will talk about it for a while. And then we have bacteria, which we will not talk so much about, but they are also part of the story and they participate there. We shouldn't forget that they are there. Of course, as anyone of us takes rather chocolate than bread, the bacteria also prefer easily available compounds, but some of them can even degrade biopolymers. And nowadays, the view of soil community, composition, function, whatever, change with the methods. And I would just briefly introduce that we can rely on nucleic acids. The nucleic acids make a report of what is there for functions, but they can also tell us which functions are important because they are expressed. So when we have a community in soil, we typically see there hundreds or thousands of bacteria and tens to hundreds of fungi in one single gram. They are mixed there. It's complex and they are in different abundances. But if we extract nucleic acids, it gives us some idea who is there. We can use a molecular barcode, for example, ITS for fungi, and to count the members of the community to see who is there and how abundant they are. We can also look at all the genes and see what functions are there. But we can also be more curious and ask which functions are essential now at this moment or in this ecosystem. And that is where we go for RNA, for the genes that are expressed and which give rise to enzymes and functional molecules. So if we look about fungal communities, they are somehow structured by the environment and this connects to their function. So partly they respond to the vegetation and chemistry of the system. This is an example again from Křtiny, where we studied the monocultures of the three trees and we found that many species are quite specifically bound to one or two of these trees, but not all. So this is one of the structuring factors that we need to consider, that the fungi somehow reflect the diversity and composition of the forest. Then there are some finer structuring factors. I mentioned the chemistry, but it's also the stand age for ectomycorrhizal in soil, in the rhizosphere, but also in the roots. So for all of them, it was not a large part, but some significant part still was associated with the age of the stand. It's again the Křtiny forest north to Brno, where we did the studies and we had spruce and beech stands from 1 to, let's say, 130 years. And with the age, the community also somehow differs because the seedlings support a slightly different ectomycorrhizal fungi than the mature trees. Then we should also keep in mind that it's not static. The fungi are not there like in a static state where they just exist as trees exist for a long time. The fungi and bacteria are more dynamic. The microbes are more dynamic. They face the differences in nutrient availability, so their communities change in time. So in one of the experiments, we asked how much they change in time, and indeed, we can measure the speed of the turnover, the speed with which the fungi are different each year. You can see that if you look at the soil fungi, it's a red line. The more distance in time, 1, 2, 3, or 4 years, the more different the communities in the same place are. So we have some idea of what change there is in time. For the litter, it looks like there is no change. Litter fungi, this is the yellow line, but this is something very different. The litter is so dynamic that we are not able to measure it anymore. There is still the same pool of fungi. So every year with the new freshly coming litter, the community actually regrows in a very different way, and there is no observable trends. For the bacteria, the trends are somehow similar to fungi. And we can also look at what's happening with soil carbon if we ask who of the microbes is using it and in which way it's using it. So if we apply labeled carbon for example, labeled plant, fungal, bacterial biomass, or individual compounds like glucose or cellulose. We can mix it with the soil, and then we can have 3 kinds of answers. We can see how much is used for energy generation, for respiration. It's one of the outputs that we can have. We can see how much incorporates into the biomass. If we measure the biomass as PLFA, the PLFA can be analyzed and distinguish fungal and bacterial ones. And we can also go for DNA and see the enrichment of the 13C in DNA, which gives us the opportunity to separate the labeled DNA, which comes from the compounds that we add to the system, from the unlabeled DNA, and the labeled heavy fraction can be read. If we read the identity of the DNA, we can look at the species that use each individual compound, and this gives us some functional answers. So if we look at the different substrates, we see that some of them are readily used for energy generation. They are respired quite well. The others are much lower in being respired. And we can also calculate the share of the 13C label in the biomass, in PLFA, and in carbon dioxide, in respiration. In there where there is a higher share of PLFA, it looks like the carbon flows more to the biomass. In the different direction, it generates more energy. So in this case, for example, the citric acid and glucose led to very low PLFA to CO2 ratio, which means that these 2 compounds are used for generating energy, which is necessary, obviously. The biopolymers like cellulose, hemicellulose, or chitin was more enriching the biomass and relatively less contributing to respiration. And if we merge all of these pictures together, we can look what happens with the carbon that flows through the system. It enters plants. You can see the plants are a bit brownish. This brownish color corresponds to nitrogen content. So the plant biomass is somewhere in the middle. And from there, the exudates or the carbon transferred to fungi and bacteria comes as a direct link, and it can be used by anyone who is there. The share of fungi and bacteria can be more or less the same, but part of the carbon goes to the dead plant biomass. And from there, we can see the first differences. So from the dead plant biomass, there is a thicker arrow going to fungi and a thinner arrow going to bacteria, meaning that fungi can get more of carbon that ended up as a dead plant biomass. It's also because the dead plant biomass doesn't contain so much nitrogen, and bacteria have large amounts of nitrogen. So it's fungi who are main decomposers of dead plant biomass. If we look at the bacterial part of the picture, there is also fungal biomass that flows to bacteria. It's dead fungal biomass, dead bacterial biomass that flows into fungi, and the bacterial and fungal biomass being recycled by fungi and bacteria themselves. Again, if we look at the arrows, it looks like arrow from fungi to bacteria is thicker than the opposite one, which means that the fungal biomass is mainly utilized by bacteria, and bacteria are important decomposers of fungal biomass. And that's perhaps also because the fungal biomass is a bit more enriched in nitrogen, which the bacteria need. And then, of course, part of the carbon is not utilizable and goes out. So that fungi participate in that plant biomass decomposition. They use enzymes and they degrade the individual components of the litter, for example. So here you can see the decomposition of lignin, hemicellulose, cellulose, and the enzymes that are used for that. And especially in the beginning of the experiment, the ratio of fungi to bacteria is very high, and the fungi have some first 4 months where they play the major role. Later, the bacterial role is increasing with the maturation of the substrate. And we should also notice that there are specialists for each decomposition step. So the very fresh leaves are decomposed by a different community than the later term. So fungi are important decomposers. Several people asked about ectomicrons, fungi, and their possible involvement in decomposition. There was a proposition that a lignin-like or manganese-like peroxidase exists there. It's called class 2 peroxidase mostly. So if we look at fungal genomes, and these are the genomes that we sequenced recently, we can see that indeed, there is class 2 peroxidase in some of the genomes, not all of them. In some of them, in the yellow color, it's really expressed in the soils if we look at the site where the fungus is growing. But there are also quite many cellulases. So indeed, ectomycorrhiza fungi probably decompose some organic matter, probably to get rid of decomposition and to utilize the nitrogen that is there. Maybe cellulose and lignin are not the major targets. The major target is probably the humic compounds, which are similar to lignin and which retain more nitrogen. We can also look at the carbohydrate utilization in general and classify them into several groups like plant compounds, microbe-derived compounds, and whatever else. And then we can look who is actually responsible for utilizing them. So in this very complex picture, the upper picture is more important. It's the transcripts, the genes that are transcribed, the enzymes that really decompose the organic matter. And each column is one group of enzymes decomposing some kind of carbon compounds. For example, the green labeled ones are cellulases, the enzymes targeted cellulose. And if we sort the bars to those which are utilized by fungi and bacteria, we can see that it's very red, and very red means very much expressed by fungi. The blue is the color for the bacteria. So for some of the carbon compounds, the bacteria are equally important as fungi, but for many of them, the fungi are the exclusive or important decomposers. The picture below is just an example of which answer we would get if we just stick to metagenomics and look at the potential. So the potential is like biased towards bacteria, but also difficult to assess because some of the fungal genes are difficult to find. They are too complex to be analyzed by metagenomics in typical settings. And if we look how it looks in general, we can also count the genes that catalyze the individual biochemical reactions. And we can see that it's hundreds, sometimes thousands of genes in a typical forest, which means that very often there is high functional redundancy and there is hundreds to thousands of microbes that do the same job. It's not always fungi, it's fungi and bacteria, but you can see that sometimes it's tens, sometimes even hundreds of taxa that can perform the same reaction. So it's not that there is some keystone species that does all the decomposition. It's really the collaboration of many. But forests are about trees, and this should be importantly stressed. And it's not just about decomposition, it's also about the photosynthesis and transfer of carbon to the fungi. So this is a spruce, this is a pine seedling with its rhizosphere, which is relatively small. But you can see that the ectomycorrhiza hyphae extend quite well into the surroundings, and the carbon flows there being transported by the mycorrhizal network, but nutrients flow back. And this is how it works, how the carbon from trees enter soil, and this is how nutrients are trafficked to the trees. And you can imagine how important primary producers trees are. So equally important is this process. And we will see this just in a moment. So if we look what happens in a temperate forest across seasons, we can distinguish the season when the photosynthesis is on, when the temperatures are favorable and the trees are growing. Is the summer season, for example, the September is the peak of photosynthetic production for many trees. And there the photosynthates flow to the roots, and through the roots to the mycorrhizal fungi. They have unlimited access to carbon. They have to mine for nutrients for the trees, but they can also suppress the saprotrophic fungi in theory. There is quite different situation in winter. In the mountains where the snow covers the soil is insulated, the temperature can be around 0 all the winter. And if you uncover the snow, you can smell the fungi very nicely because they are totally living. It's not frozen, it's a living soil. But there, obviously, there is not so high traffic of carbon. Perhaps there is some carbon left in the starch, and it still feeds mycorrhizal fungi, but obviously this flow is not that perfect as in the summer. And this is the time when the saprotrophs get rid of this fight with the mycorrhiza, and that technically they can take bigger part in accessing the organic matter. We should also see that the habitats are different. We should distinguish the litter, which is purely plant biomass, the soil, which is like leftovers, and the rhizosphere, which seems quite similar to soil, but more rich in microbial biomass. And obviously the roots, which are really plant organs, but in many cases full of fungal hyphae, of ectomycorrhizal, and so on. So you can see that they have some genes that are same with other habitats, but some which are very distinct. And also the transcription is very distinct. You can also, for example, see that there are many genes that are present the same in the soil and rhizosphere, and indeed they are transcribed very differently. In roots and litter, the gene pool is quite similar. The overlap of these two habitats in terms of gene composition is quite high, but it's extremely different in terms of what is transcribed. There are other genes needed in litter and other in the living groups of the tree. So this is the specific habitats, and each fungus either grows in its habitat or connects the habitat. But be it both ways, the habitats are distinct. The habitats here are with different symbols, and each symbol is a bit separate, which are different colors. Each color is separate. So for the metabolome, you can see that the composition of the organic compounds are very different in the habitats. And this is also the same for the metatranscriptome, which are very different. So it's just the rhizosphere and soil where the two colors of these habitats overlap, but the litter is very distinct and the roots are very distinct. But you can also see that they are open and filled symbols, and this is winter and summer. So even within one single habitat, other genes are needed in summer and other genes are needed in winter. We can also look at the spruce roots and ask what's happening there, what's happening with the tree, and how different it is from season to season. So in this kind of graph, you can see some arrows that connect the samples from different seasons. The orange and pink ones are the winter samples, which are very similar. The end and beginning of winter is very similar in terms of microbial activity, but then the early summer and late summer are very distinct periods of the year where there is much distinct processes happening. And if we look at the transcription of the spruce itself in the roots, it's many genes that are common and that are needed all the year round, but also there is a high proportion of genes that are seasonally distinct and that change from season to season. In the spruce roots, it's most of them. If we look at the root microbes, it's less. In the rhizosphere, it's still less, and litter is the most constant environment because its composition in the spruce forest, it's not changing so much with time. So there is this transition from the very plant-centric habitats with the living roots to this dead plant biomass, which is no more so much seasonal. And if we look again at the fungi that are there, the biggest difference between summer and winter is the expression of mycorrhizal genes, because obviously the mycorrhiza has more chances to flourish in the summer where it is best fed by the roots. So the proportion of mycorrhizal genes is much higher in summer than in winter. Fungi also react to management and disturbances, and let's say that management can be sometimes a disturbance, and this is another example of an experiment in the Skinny region, which was simply a forest that was clear cut. It was cut between May and June in 2013, and then we followed for 2 years what happens in the soil. And what happens, obviously, if we cut the trees, the dead roots remain in the soil, the stumps even remain there. But with the roots, if you try to recover them, there is less and less because they are slowly decomposing. So it's one of the obvious processes that the roots are decomposing. There is also the increase of fungal biomass in the roots. Originally, it's quite little, but then it goes up to 3 times higher because the roots are a source. It's no more living. It's a source of carbon for the fungi. So it's a habitat. And if we look at the rhizosphere and bulk soil, there we see actually very opposite behavior. The fungal biomass is going down up to 40% in the bulk soil, but as much as 86% in the soil close to the roots, because if the roots are dead, they don't no more feed the fungi that are around. So in the 2 years, the mass, the amount of fungi changes. It's not just the amount, it's also the composition of the fungal community that changes with time. And these arrows show what happens with the root community, with the rhizosphere and soil community in time, from the green symbols before the clear cutting up to the blue ones 2 years later. So there is some development. And if we look, the most obvious change is that the mycorrhiza originally represent something like 1/2 of all fungi that we see there. But very soon, especially in the roots, it goes down, but also in the soil, in the clear cut, the mycorrhizal fungi disappear because they are no more supported by the roots of the trees. It's just in the 2 years when the small trees regrow, there is some increase in mycorrhiza. Interesting is the picture of endophytes. It's typical endophytes of roots. And you can see that there is a very high amount of endophytes during the first year of root decomposition, especially in this root compartment. So it's fungi that are typically asymptomatic root associates, but they have their chance in the first year of decomposition where they really do their job. They disappear and probably they infect some living root to wait there until the root is dead and when they can again use this opportunity. And we spoke about the enzyme production and fungi and whatever. So again, in this case, the fungal enzymes are responding to the management, and in the bulk soil and the rhizosphere, the enzyme activity is going down compared to the original activity in the beginning, because most of the enzymes are of fungal origin and there is less and less substrate that can be reasonably decomposed. So to summarize, what is the role of fungi in forests and how they contribute to the ecosystem processes? They have a specific place or places in the food web in soils. Let's say that there is a special place for the symbiotic fungi that work on the roots and the saprotrophic fungi mainly that are specialists for decomposition. The fungi are especially important decomposers. They are important for bulk of this complex carbon decomposition, and the forest ecosystems are dynamic. It's dynamic on a long scale, but also seasonally in our temperate forest. We shouldn't underestimate it. We typically have some sampling season during the summers, but we shouldn't forget about what's happening in winter. And essentially the seasonality is driven by the activity of the vegetation of the trees, because this is the major source which makes the difference. And obviously, fungi respond to management. And if you imagine the large clear cuts or large void areas where the killed trees were recently harvested in Czech forests, which slowly overgrow with some grasses or robust plants, it can be that there the share of mycorrhiza is getting to its limits. And I can mention that we have some very recent samples from the sites after the bark beetle infestation, and it's really samples where you don't find one single sequence of ectomycorrhizal fungi. I was totally surprised that in the forest land, which will be forest once again, the ectomycorrhiza under certain circumstances can totally disappear. But what it brings to management, it also brings the notion that if we use the clear cutting, we are risking because we are posing these bottlenecks with the mycorrhiza, and maybe some retention harvesting or at least some trees left in the site would be more favorable to retain the diversity of the mycorrhiza. So thank you very much for your attention, and I'm looking forward for questions.
So, Petr, thank you very much indeed. Very interesting talk. So we will take questions in the chat box. Start it off before the first one's typed in. I think this seasonality is incredibly interesting. As a general sort of pattern, how much do you think our view of microbial ecology is biased towards summer?
Well, it depends on how you pose the question. If you would ask how much of the processes we see, I would say that with the summer we see the majority of the processes, because if we study, for example, the rate of decomposition or mycelial production in growth bags during the seasons, the winter is a really low activity time. Let's say that the decomposition activity can be 30–40% of the summer, and the regrowth of mycorrhiza can be even less. So with the summer, we capture the bulk of the activity, but obviously not the whole picture. And there can be some, as you have niches with different nutrients or habitats, it can be also niche in time. It can be fungi that just wait when they are left to do their decomposition, when the pressure of the ectomycorrhiza is relieved, and they can do their job in winter. Maybe one interesting remark, we recently have a seasonality data from Finland, and it looks that the communities there are much more dynamic. And it's because the snow comes after the frost. If the soil freezes and it's full of water in the peat forest in Finland, it really damages most of the mycelia, and then the recolonization starts from the deep substrate, and from recolonization, and the dynamic from year to year is absolutely high compared to our conditions. So maybe the picture that I presented for temperate forest is not very similar for all the temperature or seasonal effects, but it's the one that we observe in our still snow-covered forests.
Okay, related to that, we now have a question in the chat. I'll take the first one that's most closely related. That's from Michelle Scherer-Lawrence.
Fungi in roots that I presented, the fungi in roots are really the fungi in living roots. So it was the roots that were sorted, and only the living were taken. So essentially, it was not only fungi in roots, they were also fungi on roots, because all mycorrhizal root tips were counted towards the roots.
The next question was, did you find arbuscular mycorrhizal fungi after clear cutting, an increase in ectomycorrhizal fungi after clear cutting?
Yes, this is an interesting question. Actually, the growth of ground vegetation in this specific situation was not very fast, so it was very low abundance, and we didn't screen. But we have a forest where the ground vegetation is really high, and there indeed the AMF is flourishing after clear cutting or even thinning. So yes, when there is the vegetation, AMF vegetation coming, the AMF is increasing after cutting.
Okay, the next one is, how does the soil microbial community change with climatic extremes, for example, drought and heat and inundation?
Yes, this is an important question, and this is not a simple answer. I would say that fungi are quite resistant towards short-term disturbance. So if it is 1 year drought or one season of drought, the communities are quite the same. We happened to have a very dry summer one of the years. It was so dry that we decided to repeat the measurements next summer, and we saw still the same community. There was not a bottleneck effect, no exclusion. However, I would guess that if there is a pressure in time, like for several seasons, then it is like more and more probable that the fungi will be outside their limits. And if we look at the traits of fungi, for example, we map the fungi globally and look at the climatic regions that they inhabit, they indeed have some very limited space, especially ectomycorrhizal fungi have very limited space where they occur. So I guess that if the temperature changes for a long time, some of them will no more be competitive and others will replace them. So short term, I think the effects are relatively limited. In long term, if it is a development, drying or higher temperatures, then I think we will see the change in community.
Okay, the next one is what different behaviors would you expect between clear cutting and retention forestry?
Yes, I can say that actually in a retention forest, 2 years after thinning, we see similar fungal biomass and similar composition as before the operation. The only exception is the year of thinning, where there is some stress, and the share of ectomycorrhiza and fungal biomass goes down, but just for a limited time. So even if you cut like 60% of the trees, you can retain most of your ectomycorrhiza there. In the clear cut, there is always some drop. It's not always drop to 0. It's sometimes drop to 10% of ectomycorrhiza coming from 50 to 10. But this is some permanent situation. And I would guess that the recolonization is quite good, but in a stressful situation or in large clear cuts, it can be a limitation. So I would be in favor of using more of this retention forestry.
And a related question to that, which is what proportion of trees should be retained if we're going to have a retention forestry?
Well, this is where I don't have any answer. We now have experiments running, so I will need to, when they are finishing, because there are different levels of thinning, but I think it's relatively few. I think if there is something retained, at least I would guess 10, 15, 20%, that would be good. And for example, the rotation they used in Slovenia, where they cut beech forest in 3 instances, always collecting 1/3 of the major trees, that works perfectly. So I mean, removal of 70% of trees should still work. It's partly also about climate. If you remove too many trees, maybe the heating of the soil surface is different. But clearly, my guess is that you can cut relatively much, but I don't know how much, because we don't have an exact answer.
Okay, one to be decided. The next one leads on from another question from Andrea Pollard, which is, I can imagine that the fungal composition is similar. You must have been talking about this. But what about the transcriptome after drought? So what happens to the transcriptome?
That's a difficult question, and it's also technically difficult, because essentially, if it is dry, the transcription is slow, and you can recover very little RNA. So there is no scientist that really studied transcription after drought, because technically it's difficult to get RNA. When I started my research career, the RNA was an imaginary molecule that no one could get from soil. And people told me, just go after rain. It's the only situation when you can get RNA. But I can give you an opposite answer. We applied watering to dry soils and compared the controls and watered forest soils and wanted to ask what's the difference after the rain. And the answer is there is no difference. Everyone likes water. All of them are expressing almost the same as in the control. So it's just a boom of activity, very high respiration, but all of them are happy after rain. So this is the opposite answer. I don't have the draft answer.
Okay. And then we have one more question. How close are we being able to classify saprotroph fungi or ectomycorrhizas or bacteria into more specific functional groups?
Yes, well, I think that we have some understanding of what these groups can be. That's one thing. And for example, within the saprotrophs, we can distinguish the ones that can really cope with complex substrates like complex hemicellulose or lignin or whatever. And then the molds that are just opportunists, can decompose just very simple disaccharides, whatever, easy molecules, and are waiting for the exudates. So this is a distinction that we can make. I can't say that we always know who belongs where. For bacteria, we actually classify them into those who can and transcribe decomposition-related genes and the so-called opportunists. And it's interesting to see that the opportunist, which is, for example, many Burkholderia species of bacteria, they have like 10 times higher abundances in summer when the roots exude and there is this carbon flux in the whole of the soil. In winter, you don't see them. While the acidobacteria that are decomposers, you can see them in litter all year because they can decompose the cellulose themselves and feed there. So it's two distinguished groups, but we are very far from sorting each individual bacterium or fungus into these groups. So we know where to go. I hope that our experiments will let us know. And then another classification can be based on the preferred substrate. So for example, we incubated different substrates and looked at the fungi and bacteria that prefer dead fungal biomass, high tin, cellulose or lignin or whatever. So this is technically easy to do, but to classify the diversity, it's difficult. If you can imagine that it's really tens of hundreds of species that do almost the same, it's very difficult to classify them all.
Truly fascinating. We have probably one last, which is from Cindy Prescott, which says that on the soil sea flow slide, you show N content of bacteria and fungi are lower than in plants. Is not the opposite true?
Yes, probably I made a mistake, but or maybe it's just a misunderstanding. The nitrogen content in bacteria and fungi is higher than in dead plant biomass. Like the C/N ratio in dead plant biomass can be 100. In bacteria, it's like 14, and in fungi it can be like anything between 10 and 40. So nitrogen is more concentrated in microbial biomass than in dead plant biomass. Maybe there is a mistake in the picture.
Okay. Well, the answer to the reaction is pew. So, okay, we'll take two last questions. We've got a bit of time. How does the arbuscular mycorrhizal fungi versus the proportion of ectomycorrhizal fungi change with soil depth? And what would be the implications for forest resistance to drought?
Well, I think that essentially we don't have the data because we don't analyze arbuscular mycorrhizal fungi very often. They require different methods and they can't be so easily measured as the ECM because they require other primers and they have other markers. But I can say that even in grasslands, which is a fully AMF plant system, the AMF fungi are relatively small proportion, maybe 10%, and most is saprotrophs. So in forest, I expect that it's very little share of AMF and that they actually don't affect the ECM, that there is not so big fight because the biomasses are very different. With the depth, I would guess that ECM goes much deeper because at least for the beach, the roots go deeper. So that would be my guess, but again, I don't have an answer. And
Okay.
the implication for draft, I can't say.
Fine. That's what the seminar is all about. So another question, a very interesting question was, as you said that after infestation with bark beetles, there are no, or the ectomycorrhizas goes down. The question is this due to a change in the contribution of carbon from the host to the fungi, i.e. starvation because there's no needles? Or could it be that it's also an effect of the host defense system being deployed to resist bark beetles?
Well, I must mention that what I mentioned was an extreme, that it's not all post-bark beetle sites that are devoid of mycorrhiza. I should say that even in the clear cuts, we observe that the retention proportion of ectomycorrhiza is quite different among sites. There where there is no ground vegetation, like beech forests in the mountains, much of the mycorrhiza is retained even in the clear cuts, it's just probably not active. While there, where there is a regrowth of robust plants, grasses, all of ground vegetation, the ectomycorrhiza is probably easily wiped out by the new symbionts of the new grass layer. So this is, to me, the biggest difference. But we have too few data to say. But I don't want to be misunderstood. There are post-barbecue sites where the ectomycorrhiza still exists, there where the regrowth is not that fast, or where the grasses don't overtake. It depends on the nutrients in the soil.
Okay, one last question. What would happen if you do not harvest the dead trees, presumably in terms of this?
It's the same. We studied post-barbecue sites in the Bohemian forest in the national park, and the trees were retained there, and it's the same. The ectomycorrhiza goes out. They are no more supported, and for some time they are retained, but it's quite the same. So no matter if you remove the trees or leave them, the bark beetle like wipes out the productivity of their symbionts, and this is lethal to them.
Okay, so Petr, now thank you very much. Thank you very much for all the people with the questions. Some really interesting questions. Petr, thank you very much for a very, very interesting talk. Lived up to all the expectations. So once again,
thank you
very
for
much.
the invitation, and I would be happy to see you face to face sometime at the conference, maybe on ICOM or IMC. So
yeah, that was the reminder that ICOM, which we had at the beginning, you can still submit an abstract.
Yes.
And lastly, a reminder for everybody that our next seminar is on the 5th of June, and it will be Luke McCormack from the United States. So see you all on the 5th of June. And thank you very much for coming.